Hi everyone, this recorded lecture covers anatomy of the special senses. And we're going to start by looking at taste, more appropriately referred to as gustation. Our primary taste sensations include many things you could already guess, I'm sure, such as salty, sweet, sour, and bitter. Umami might be a new term for you. This refers to savory. That's savory taste sensation that you might get when you're eating such meals as a turkey dinner. Taste or gustation begins with the chemical stimulation of sensory cells that are clustered in about 4,000 taste buds. The chemical stimuli, though, are called tastings. Most taste buds are located on the tongue, but some do occur on the inside of the cheeks, on your soft palate, in your throat. We're going to have a look at lingual papillae on the next slide, but lingual papillae are those bumps that you would see on someone's tongue. Taste buds are groups of taste cells. And on upcoming slides, we're going to look at all these different terms. Let's start with the first one being lingual papillae. Papillae really means this elevation. If you think back to talking about dermal papillae, it was those projections. Lingual refers to the tongue. We have filiform, foliate, fungiform, and vellae lingual papillae. Filiform, which are not explicitly identified on this image, these are scattered throughout the tongue. They do not have any taste buds within them as part of them. It is the filiform that do allow you to experience different textures of food. It also gives a bit of roughness to our tongue. Cats have a lot of filiform lingual papillae that contribute to the roughness of a cat's tongue. Foliate, these are considered to be more weakly developed, and they actually degenerate by age 3. So they're really prominent when a child is young, when you are young, and they are found on the sides of the tongue. Fungiform, these type of lingual papillae contain a few taste buds, and they're found at the tips and the sides of your tongue, so really through here. And finally, vellae lingual papillae. These are found at the rear of the tongue in really a V-shaped form, and these are large. They contain up to a half of all of your taste buds. So this is the big one then of all the lingual papillae for taste buds. Let's have a look at taste buds. These take on a lemon shape, and you can see it in this image here. And they consist of groups of about 50 to 150 different cells, including taste cells, which we can see here, supporting cells, which we can see here. So the blue is the supporting cell, the taste cell is purple, and basal cells, which are really at the basal end of the taste bud. Now, let's look at the taste cells themselves. So again, we're looking here at the purple structures, the purple cells. You can see that they consist of microvilli at their apical surface. So if this is the basal surface, here's the apical surface here. And this region here, these microvilli, they're taste hairs, and they serve as the receptor for taste molecules. So when you eat something, it will dissolve within this region, the molecules of what you eat, or those tastants, in order for us to interpret taste. We also see a taste pore. So this is this porous region here where the taste hairs do project, but this is also where food molecules will dissolve. These taste hairs are not neurons, they are actually epithelial cells. And if you follow the hair here, which we know is attached to the taste cell, to the basal part of the cell, you can see that there are these synaptic vessels that contain neurotransmitters. And so when we have a food substance that dissolves here, it will signal these synaptic vessels to release their contents, which will then come into contact with these sensory nerve fibers that are found at their base in order to transmit this to our brain for us to interpret taste. Now, we also mentioned basal cells, so let's actually go back for a moment. I said that taste buds are made up of taste cells, which we just spoke about, and supporting cells and basal cells. So let's have a look at the basal cells and supporting cells now. The basal cells are the stem cells, and these are going to replace every 7 to 10 days, which is actually quite interesting. So any cells, taste cells that have died can be replaced every 7 to 10 days. And they may have a sensory role as well, just because of their location and proximity and the synapses that have been identified with the sensory nerve fibers. Supporting cells, that third cell type here, the blue cells, you should note that they do not have taste hairs present. You can see that if you look at them closely, so no microvilli at their apical surface. And they do look quite a lot like taste cells, but they also don't have the synaptic vesicles present with neurotransmitter, and they also don't synapse with the sensory nerve fibers, so they're not really playing any role in sensation. But what they do is, as their name suggests, is they support the taste bud. Now let's move on to smell. So we just discussed taste, and we're going to have a look at what allows us to interpret the different odors around us, and the appropriate term for smell is olfaction. Our nasal region consists of olfactory mucosa, which contains 10 to 20 million olfactory cells. Now, olfactory cells are neurons, and you might remember from the previous slide that taste cells are actually epithelial cells and not neurons. So that's certainly a difference between these two senses. But there are also supporting cells, just like with taste, and basal stem cells, just like with taste. Now, this mucosa that's involved in smell is found on the roof of our nasal cavity. So if we have a look at this sagittal cut right here of the face, here is your mouth, your tongue. This is the hard palate or the roof of your mouth. And here is your nose where you would breathe in odors. And right at the roof here, this is where your olfactory mucosa, or the mucosa involved in smell, is going to be found. Now, at the top here, you can see a bone. And above this bone is where your brain would be. And so we're going to have a look on the next slide, really what's involved in this region right here between our nasal cavity and where our brain is, or between that, our nasal cavity and our cranial cavity. And if we look closely, you can see the brain here. So we're zooming in here, down here. And you can see the brain here. You can see now that there's nerves here. Here is that roof of your nasal cavity right here. And then your nasal mucosa, olfactory mucosa here. So we'll have a look at this on the next slide. But on average, we can distinguish between 2,000 and 4,000 odors. So olfactory cells, as we've already talked about, are afferent neurons. And the great thing about these types of neurons is that they have the ability to regularly regenerate compared to other types of neurons in the body that we've said cannot. And they take on this unique shape, the olfactory cells that is, where they look like a bowling pin. And you can see that in this image here. It looks like an upside down bowling pin. And it's described as being this modified dendrite with a swollen tip. So you can see an axon here, you can see your neurosoma or cell body here, and the dendrite, which doesn't look like your usual dendrites. You can see instead it's like got almost a long neck on it, and then this swollen tip, which is like the head. So think of it as almost somebody, well, and you have these olfactory hairs here, so it's almost like somebody is standing on, doing a headstand. And so these olfactory hairs then are going to be greatly involved in the dissolving, or the binding, I should say, of odor molecules that allow us to interpret smell. They're considered non-motile, these cilia, meaning that they don't actually move. Now the basal end, so this would be the basal end here, is where the axon actually arises from. Now let's look back up at that roof here of the nasal cavity, and then this would be really the floor of the cranial cavity. So we have the roof and the floor, and this is actually your ethmoid bone here. And your ethmoid bone has these holes running through it that really connect the nasal cavity to the cranial cavity, called the cribriform foramina. Foramina means hole. So there's all these holes that are present here that will allow for the passage of the axons from your olfactory cells. And so what we can envision maybe here is that groups of axons are going to come together, or nerve fibers, just as we know they do, which will form a fascicle. So we've talked about that before. So we have these groups of nerve fibers that form a fascicle, and they will collectively then penetrate through the cribriform foramina and become part of your olfactory nerve. And your olfactory nerve is what will then bring information to specific regions of your brain that allow for you to interpret an odor. So let's see if we can almost take this entire structure here and place it down here. And then we'll have a whole bunch of them come together. The axons will form a fascicle, and they will penetrate through this cribriform foramina, becoming part of your olfactory nerve, transmitting the sensation of whatever odor you've breathed in, represented here. And then you'll interpret it as to what that smell is, but also whether or not it's something that you like or don't like. Now we'll move on to hearing and equilibrium. Hearing, our ability to hear, is a response to vibrating air molecules. So we call this a form of mechanoreception, as we talked about mechanoreception at the beginning of this lecture. Equilibrium is really your sense of where you are, where your body is in space, whether you sense motion, your ability to sense balance, to kind of orient yourself in space. And both of these senses reside within your inner ear. And our inner ear is this maze of fluid-filled passageways and sensory cells, as we'll see coming up. And what will happen, just speaking very generally, is that fluid within the ear will be set into motion, and sensory cells found within your ear are going to convert this motion into an action potential or an impulse that will allow for you to interpret sound. Let's start with some anatomy of the outer ear. Here we have the outer region of the ear that we call the auricle, or the pinna. And the pinna is what is really going to be directing sound into your auditory canal. So here's your auditory canal. And so the pinna here, or the auricle, will almost funnel or channel sound into your auditory canal. And this canal is a passageway through your temporal bone. So here's part of your temporal bone here, and here's a passageway through the temporal bone that developed when in utero. And this will allow you to get to your eardrum, which it will be shown on an upcoming slide. And the eardrum is more appropriately called your tympanic membrane. Within our outer ear, we're going to have various hairs that are going to be here that help protect your canal. There's also going to be the production of cerumen by ceruminous glands, which is your earwax, again acting as protection, trapping foreign substances that shouldn't be there. Now we'll move on to the middle ear, and the middle ear would be us moving on more medially. Here's your auditory canal, which takes us to our middle ear. So middle ear is this entire structure really here. But the ones to really point out that are structures that are found within the middle ear include the tympanic membrane, which is your eardrum here, sort of a drum shape, and your tympanic cavity, which is just a little bit internal to that, which is an opening here. The eardrum itself has this ability to vibrate freely in response to sound. It's innervated by two cranial nerves, the vagus nerve and the trigeminal nerve, and it's very sensitive to pain. So if hearing loud sounds, or if you've ever put a Q-tip in your ear and hit a part of your ear, or had water trapped in your ear, you've experienced probably a lot of pain when this happens, and that's because of its innervation. The tympanic cavity that's found behind it. This is really the region that's between your outer ear here and your inner ear here, and it contains your auditory ossicles. So these small bones, the smallest bones in our body, the stapes, the incus, and the malleus. And it's these structures here that really connect your tympanic membrane or your eardrum to your actual inner ear over here. So here's showing you again those auditory ossicles found within your middle ear. Another structure to point out is called the auditory or eustachian tube. So if you follow your tympanic cavity here down through here, we have an auditory tube. Please don't mix up your auditory tube from your auditory canal. Sometimes I even mix up those words when I speak, so just make sure you know that this is the canal here leading to your ear, and here's the tube that's also known as the eustachian tube. And what this tube does is it connects your middle ear, so this region here, to your nasopharynx. And you'll learn in later lectures that the nasopharynx is the part of your throat, pharynx means throat, that's located behind your nose. So there's a connection then between your nose and your ear, which probably makes sense to you, because if you're sick and you have a sore throat, then it can turn into ear pain, and ear infection or ear pain can turn into a throat infection as well. But the purpose of this tube is to equalize air pressure. This is what feels blocked when you are ascending and descending during a plane ride. And so some people will plug their nose and apply and sort of blow through their nose while their nose is closed to open up that auditory tube, because it is normally closed, and so opening it up helps to reduce pressure that you feel within your ear. Swallowing and yawning or chewing gum on a plane can sometimes do that as well. This is showing you here the eustachian tube of a child versus an adult, and you can see the connection then with this auditory tube between your ear and your nose, and so this would actually connect back to your throat as well. And the auditory tube in a child is more horizontal and shorter than it is in an adult, which can predispose babies to ear infections and poor drainage, or children as well. Now let's look at the inner ear. The inner ear is housed in a maze of temporal bone passages that are called the bony or osseous labyrinth, and it's lined by a system of fleshy tubes called the membranous labyrinth. So the bony tubes are lined by membranous labyrinth, or just lined by a membrane. Now between the bony and the membranous labyrinth is a cushion of fluid that's called the perilymph. And the perilymph is similar to cerebrospinal fluid. Now within the membranous labyrinth is a fluid called the endolymph, and this is similar to fluid that's found within cells. The bony and membranous labyrinth form a tube within a tube structure, somewhat like a bicycle inner tube that's found within a tire. And on this next slide, you can see an image of sort of what that looks like, the tube within a tube structure. So the different linings that exist, or the different passageways that exist within the bone. So a bony labyrinth of passageways lined by a membranous labyrinth. In the inner ear, we have a vestibule and a cochlea. The vestibule is our organ that's involved in equilibrium, and the cochlea is the organ of hearing. And if we put these terms together, the vestibule cochlea, or we could say the vestibular cochlear nerve, you might recall that that's a cranial nerve involved in both your equilibrium and your hearing. And so the vestibule is really this region through here, and we're going to talk about this on an upcoming slide, but your cochlea is located here. You can see your cochlea as well located here, and the vestibule located here. So let's have a look at this on our next slide. So let's have a look at what's called the vestibular apparatus that's involved in our equilibrium. We talked about the vestibule just briefly on the previous slide. So what is equilibrium? It's our ability to be coordinated, have balance, orientation within a three-dimensional space, and it can either be static or it can be dynamic. And when we talk about static equilibrium, this is the perception of the orientation of your head when your body is stationary, whereas dynamic is the perception of motion or acceleration. Now, your vestibular apparatus has receptors for equilibrium that consists of 3 semicircular canals or ducts and 2 chambers. So let's see what those look like. We have the 3 semicircular ducts here, anterior, lateral, and posterior. And they detect angular acceleration. I'll talk about that in a moment. The 2 chambers are called the saccule and the utricle, and these are responsible for static equilibrium and linear acceleration. Now, what's the difference between angular and linear acceleration? Linear acceleration is a change in velocity in a straight line. So, for example, when you're riding in a car in a straight line direction, or even riding in an elevator. Whereas angular acceleration is a change in the rate of rotation. So let's say, to make this more simple, that when you're in your car and you're experiencing linear acceleration, but then all of a sudden you decide to turn a corner, that would be experiencing angular acceleration, which our 3 semicircular ducts are involved in. Or when if you're rotating in a chair, in a swivel chair, and you're swiveling or rotating around, that would be a type of angular acceleration as well. Okay, light and vision. So the eye is our last topic for the sensory system or sensory organs. We're going to talk about vision and light. Vision or sight is our perception of objects in our environment because of the light that they emit or the light that they reflect. And in order for us to actually be able to interpret what is in front of us as what we see, is that light that is emitted or reflected must cause a photochemical reaction in order to produce a nerve signal. Just like every other, that nerve signal is important for any form of interpretation, as we've discussed. Light and vision is considered our dominant sense just because of the number of receptors that are present. Let's look at some accessory structures of the orbit. We have the conjunctiva. The conjunctiva is this transparent membrane. It's a mucous membrane that lines your eyelids and the anterior surface of your eyeballs. So really underneath through here, your eyelids, and then the front part of your eye, with the exception of the very front here, which is the cornea. So except the cornea, and we'll talk about the cornea shortly. The conjunctiva has a lot of nerves. It's very vascular. If you've ever had pink eye before, what you've had is a form of conjunctivitis, which means inflammation. Itis means inflammation of the conjunctiva. And your eye's probably hurt and has probably been bloodshot as well, and that's because of the innervation in vascular supply. What's the purpose of this conjunctiva? It's going to secrete a thin mucus that helps prevent your eyeball from drying out. We also have a lacrimal gland, which you can see found on the superior lateral part of your eye. And it has these ducts that run through here. And what will happen is you produce tears here that travel through these ducts, will wash through your eye, and then will drain through here, through this lacrimal sac. And that's where you will either swallow your tears or they will come out your nose. We have muscles in our eye that allow for movement of our eye in the many different directions. We have 6 extrinsic muscles. These are meaning that they're on the outside of the eye versus intrinsic ones that would run and be within the eye. We have 4 rectus and 2 oblique muscles. 4 rectus muscles include superior, inferior, medial, and lateral, and that just infers their location. So it's either found superiorly, inferiorly, medially, and laterally. And they're called rectus, just like your rectus abdominis and your rectus femoris, just because of how those muscle fibers tend to run. And they're going to move your eye in the direction that you would expect. So if it's superior, if you can envision the superior rectus muscle here, if you can envision this shortening and contracting, then it pulls your eye up. If you can envision this one here shortening and contracting the inferior rectus, it's going to pull your eye down. Same with the lateral rectus. If it shortens, it's going to pull your eye laterally, and the medial rectus will pull your eye medially. We have oblique muscles too. They run obliquely, hence their name. And we have superior and inferior obliques. Now, each of these muscles actually have multiple different directions that they move your eye, but we could really just say that the combined vector action of the superior oblique is down and out, and the combined vector action or the vector summation of your inferior oblique is going to be up and out. The next handful of slides are going to be talking about the different layers, the different components of our eye. And so this is a summary slide that I suggest you come back to in order to summarize what we're discussing on the next few slides. The tunics of the eye refer to the different layers of the eye. And you can see that we have an outer fibrous layer called the tunica fibrosa. So it is a layer that's made of fibrous tissue, and it's the most outer. We have a middle vascular layer called the tunica vasculosa, also called the uvea, and it gets its name vascular because of its heavy vascular supply. And the tunica interna is the most internal. So let's break them down. This outer fibrous layer, so the most outer part of the eye, consists of your sclera and your cornea. The sclera is the white part of your eye that extends even to the back part here. So here's the sclera. And the cornea. So if we follow the sclera right around to the front, now we have this modified form of the sclera at the very front of your eye that's called the cornea. And it's going to be the cornea that actually allows for the admission of light into our eye. Moving more internally now, we'll look at the choroid. So the choroid is this brown region. And it's brown because it's heavily pigmented and it's highly vascular. And you can see the vascularization in this region that's showing you, it's cut back a bit to show you the different vessels that are present. We also have the ciliary body. So let's follow the choroid here to the front. And now it changes a bit. This is called your ciliary body. So it's really an extension of the choroid where it's a muscle. And it's a muscle that's going to support your lens and support your iris. So you can see that the lens here is supported by the ciliary body rather. And even through here, the muscles that make up part of your iris are also supported by the ciliary body. In addition, the ciliary body is going to secrete an aqueous humor. Aqueous humor is a fluid produced by the eye that provides the eye with nutrition, but it also helps maintain the eye's pressurized state. So if you've ever had pressure checked within your eye, which is something frequently checked for a condition called glaucoma, what they're doing is they're measuring the pressure that's being exhibited or given by the aqueous humor. We also have an iris. The iris is that colored part of your eye that's part of our middle vascular layer, the tunica vasculosa, and it's the iris that contains muscle. So when it's cut, you can see the muscle through here that's either going to tighten and constrict tighten and constrict or dilate and widen to change the size of your pupil. So if you're in a dark room versus a bright room, your pupil size is going to be different. So let's say we're in a dark room, your pupil size is going to be larger because there's less light available, so it has to open up more to try to get more light in to allow you to see as much as possible. And then if all of a sudden somebody turns on a bright light, well, if your pupils are still large, then it causes your eyes to be very sensitive and you'll squint and your eyes will hurt a bit. If you're in a bright room, your pupils are going to be a smaller size because there's so much light available that they don't need to be as large. Now the tunica interna, the most internal layer, consists of your retina here, the most internal layer. So you could see the layers here, 1, 2, and now we're on the third internal. So your retina and your optic nerve here at the back. And we'll talk a little bit about this shortly. The optical components of your eye are these elements that are going to emit light rays. They're going to bend or refract them and focus the image on your retina, which is going to allow for interpretation of what you see in your environment. The cornea we described already. The aqueous humor we discussed as well, and the lens, but what's new to you is the vitreous humor. And the vitreous humor, or the vitreous body, is a jelly substance that makes up a good portion of your eye. So even if we go to this previous slide here, your vitreous body is all going to be within here. And it's a jelly-like substance. And it has multiple functions, but what it really does, all of this jelly fluid that's found behind the lens of the eye, so once again, all through here, what this is going to do is allow for your eye to maintain its spherical shape, keeping it round, because of all that jelly that keeps it in a round spherical shape. The neural or nerve components include the retina and the optic nerve, which we said are part of the most internal layer of the eye, or the tunica interna. And it consists of the macula lutea, which if you look here, so here's the macula lutea. If we look at the back of the eye, so when your eye doctor looks in your eye, they're looking in part at the very back of your eye. So this is showing you the back, or the fundus of your eye. And if we look at the back, we can see important structures, including the macula lutea. And this is just a patch that you can see here. The fovea centralis is the central part of the macula, that denser region and color that you can see. And this is where fine detail is going to come up for images, because it's packed with receptor cells, allowing us to see these fine images or create the vision that we see when we're looking at fine detail. And then finally, the optic disc. The optic disc here is where your optic nerve is. So let's look here. Here's your optic disc, which would be this region here, and that's where your optic nerve is located. So here's your optic nerve, where your optic nerve enters your eye at the optic disc. And so here you can see it in a different way. So we're looking in the eye at this direction. You can see the optic disc here. And this also shows you can see all the many vessels, but this is where your eye doctor can look at nerve health as well as vascular health. They can have a look to see whether or not these vessels are functioning as well as they should. It's considered a blind spot. This region here, this optic disc where your optic nerve comes out, actually doesn't have any receptor cells that contribute to vision. And on our final slide, we're just going to talk briefly about rods and cones, which are the photoreceptor cells that allow us to see. Rods are going to be greatly involved in your night vision, allowing for this grayscale vision, monochromatic, meaning using only one color. And then cones allow for your day vision, so these are going to be big in color vision. I'd like to point out here that we have many more rods as part of night vision than we do cones that are big in our day vision. And you can see what they look like in this image here, these photoreceptor cells. We have the rods that actually take on a rod-like shape, and then cones that take on a cone-like shape, hence the name that they were given. That's the end of this lecture. Thank you so much for listening.